at Harvard University in Cambridge, Massachusetts, United States. I am not in my favorite city in the world, which is where you live. Well, actually, no, you're at Sabancı, so you don't quite live in Istanbul. You live somewhere close to Ankara, or yes, whatever, Izmit. But anyway, no, I love visiting your city. I love visiting your university, and it's kind of sad that I'm not there now, but it is actually kind of cool that we can nevertheless do this. The marvel of bloody Zoom, as we call it. Like you, everyone else over here in the US is a little bit fed up with Zoom, but it does have its advantages. What I'm going to do is dive straight into my presentation. Let me just launch that. Good. So, yeah, what I'm talking about is something that I hope you agree with. Well, maybe you don't agree yet, but I hope at the end of this talk, you'll agree with me that this is a truly exciting area of endeavor, a truly exciting area of science, and it's absolutely at the heart of biology. What I'm interested in is determining what features of our genomes, human genomes, fruit fly genomes, everyone's genomes, bananas genomes, have been determined by the action of natural selection. So my title, as you can see, is Darwin and the DNA, Detecting Adaptive Evolution in Genomes. So here we have some DNA with some mystery letters in there. And lo and behold, what do we have? Excuse me, there we go. Oh, it says the origin of species. So we have found Darwin in the DNA. So yes, he's there. The question is, and this is a technological question, a methodological question, is how can we identify the key events in molecular evolution, the key events that have made us what we are today, bananas what they are today. And I'm going to start with this because it's my favorite clip of an amazing adaptation. What has made spiders the way they are today? This is a particular kind of spider.
She reaches down with her forelegs to check how far away she is from the ground.
That traps her prey with a very special
device. She snips most of the framework threads and holds the fuzzy rectangle between her 4 front legs. She's ready.
We're about to go into slow-mo.
Her enormous eyes are so sensitive she can hunt in near darkness. A bush cricket would make a rich meal, but it's very powerful and it could put up a good fight.
Boom! Now, I don't know about you, but I think that is absolutely amazing. Think about it. That spider has constructed that net, that specific net, for a particular purpose and deployed it in exactly the right way. That spider didn't buy that online at Amazon. The spider didn't somehow pick up this information from other spiders. No, all the information that created that piece of behavior you've just looked at is encoded in that spider's genome. So there are mutations in the history of that spider's lineage which have gradually created and perfected that behavior to create the perfect web net, to create the perfect distance between the spider and the ground. And then obviously the actual skill in deploying that net and using the net to catch the insect, its prey. That's extraordinary. And that is adaptive evolution. That is Darwin and the DNA, if you like. That's where that came from. There were mutations, beneficial mutations, which made that behavior ever more effective, stretching back over tens of thousands, hundreds of thousands, millions of years in the history of those spiders. So this, needless to say, is not a talk about spiders, but I just want to that's to get you into the mood. We're talking about extraordinary achievements by nature, that is adaptation. And of course, it all starts you know this, natural selection is a simple process. You have random mutation, random genetic mutation. Natural selection is just a sorting algorithm. The ones which are good thrive. The ones which are bad are eliminated by natural selection, and the ones which are, if like nothing, neutral, as we say, are ignored by natural selection. And actually, mutation in general is a bad thing. Because if you mess with a complex system and make a random change to it, it's not going to improve the complex system. So in fact, natural systems have evolved a great many processes to minimize, to eliminate as much as possible the amount of mutation that occurs. It's actually kind of striking, though, that what we, even with all that evolution to minimize mutation, mutation is still happening. It's a thermodynamic inevitability. This is actually a super cool piece of data from a study from just a few years ago, which is looking at the number of new mutations which occurred in you when you were born. The number of new mutations which occurred in you when you were born. And it's about 60 or 70 de novo, as we call them, mutations. And interestingly, as you can see, the blue line is your father, the red line is your mother. You've probably inherited many more mutations from dad, and that's simply because of the difference in the cell processes in generating sperm versus generating eggs. So dad tends to do more cell replications, more error, and that's what mutations are. They're generally copying errors in DNA. And also dad, as you see, keeps going longer. Women enter menopause and stop reproducing, whereas men like Rupert Murdoch keep on going forever. So we've got mutation is an inevitable input into the system. What happens to those mutations? Well, look, I've already told you, if you've got, and this is my attempt to make this a very Turkish talk, there is a Tofa Murat, right? Mutations, let's take a simple car, like your Uncle Mustafa's car, which is one of these. It's from the late 80s or something like that. It's an incredibly simple car by modern car standards, okay? But you open the hood and you make a random change to that system, what happens? You're going to break it, right? So almost all mutations in functionally important parts of genomes are going to be problematic. They're going to be detrimental to the system. So almost all of natural selection is what we call negative natural selection, which is just getting rid of the bad stuff. And as I say, 99.999% of mutations which occur in functionally significant part of the genomes are bad. So let's talk about boring, and it is boring. Negative selection, all it does is keeps things the same. All it's doing is eliminating innovation. It's not going to give us adaptation. What it does is it maintains adaptation. Here I've got two pieces of DNA. Well, I mean, it's one piece of DNA, but two kinds of DNA. And one, the green piece is useful and important DNA, and the purple piece is not important. Okay, it's performing no particular function in your genome. Now, what happens if a mutation, that's a mutation which has just occurred in an important piece of DNA, that's maybe disrupted that gene, the protein is no longer functioning, what happens? It's eliminated by natural selection. What about a mutation in the fairly low usage piece of DNA? It doesn't matter, nothing happens. Another mutation in the green area, removed. You get the picture? That every time you have a mutation in a functionally important part of the genome, negative natural selection does its job. And this is actually an extremely important observation in terms of our ability to understand the utility of different parts of genome. So here's a thought experiment. We've got the same situation. Purple is useless, green is useful DNA. We're starting with a single ancestral population. We're going to split it into two daughter populations. And what's going to happen? Well, it's just as we described previously. We're not going to allow the accumulation of mutations in the green region, but we are going to allow it in the purple region, right? Now, but they're going to be different mutations in the purple regions in the two different populations, right? Because these are two independent populations. So what does that mean? That means that over time, these two populations will diverge for the purple region, and I'm going to color code their divergence. So over time, as we're traveling through time, the purple region, well, the whole region is picking up mutations, but the purple region is not getting rid of the mutations. The green region is staying green, staying the same, because it's eliminating all the mutations. So see what happens? Over time, the green region stays identical, whereas the purple regions, the useless regions, have diverged. So this is actually a key tool in allowing us to understand the functionality of different parts of the genome. If you've got a piece of DNA which is very similar in terms of sequence, or a protein which is very similar in sequence between, say, humans and mice, then we know that's a functionally important region. If you've got a region which is very different between humans and mice, then probably that's not performing any very significant function. We can see this when we line up proteins. If we look at the right, this is just a bunch of proteins from a bunch of mammals, okay? And this is the single letter amino acid code we're looking at here. And blue means identical across all those different species of mammal. So what I want you to look at here is this block, what is it, 15 amino acids or so? What has happened over the history of evolution since the origin of mammals is any mutation which has landed in any of those lineages which changes the amino acid sequence of that region has been eliminated by negative selection. Negative selection is an incredibly important and incredibly powerful force in the history of life. My favorite example is a really almost sort of crazy extreme example. This is a gene which has two names because it was independently discovered in two different systems. In mammals, it's called PAX6. In fruit flies, it's called ILUS. It's called ILUS because if you have a mutation in that gene, you don't have any eyes, eye-less, okay? And it turns out that, and the same with PAX6, in mammals, if you have a mutation, you basically, the mammal, the baby mouse or whatever, does not develop eyes. What this is a gene which acts very early in embryogenesis, in producing the embryo, and says, build an eye here. Okay, so it evolved very early in the history of animals, maybe on the order of 550 million years ago. This is a cool thing. And it's just been inherited ever since to the fruit fly lineage and the mouse lineage. This is amazing. If you've got a fruit fly with a functionless eyeless gene, so it has no eyes, you can take the gene out of mouse, put it in the fruit fly, and the fruit fly gets eyes. That's amazing. It's like taking a part from a very distantly related thing and rescuing the function of the original one. Let's just make the point here. Here on the left, you can see basically all of the history of animals. Fruit flies and mice diverged. They had a common ancestor, probably, as I say, around 550 million years ago. So throughout that time, whenever a mutation has arisen, which has changed that gene significantly, it's been eliminated by negative selection. And we're actually talking about 1 billion years of evolution because we've got two separate lineages here. 1 billion years of carefully constraining negative selection. So negative selection is powerful and important, but it's really boring, right? All it's doing is keeping things the same. Where does innovation come from? Where do new things come from in evolution? Where does adaptation arise? How does it arise? And this is the other end of the selection spectrum. This is positive selection. So this is when a beneficial mutation, a mutation that makes the zebras run faster, arises. That will be picked up because the individual zebra with that mutation will survive at a higher probability, produce more offspring. Its offspring will have that mutation. They will produce more offspring and so on. So eventually the frequency of that mutation will increase in frequency until the entire population, the entire species has it. In other words, you go to what we call fixation, which is 100% frequency of that favored allele. So how do we identify the events in the history of life that have resulted in positive selection? Well, you might say, well, it's pretty easy. We can take genomes. Let's take us. I've got Miley Cyrus here as the canonical human being, obviously a good choice. We can take our closest living relatives, chimpanzees. We've got their genome sequence. We've got our genome sequence. We can line them up and play spot the difference. Okay. So obviously those differences have arisen in the 7 million years since we had a common ancestor. So 7 million years ago, the two lineages split. One produced humans and the other produced chimpanzees. Okay. And in that time, only 1%, so about 0.5% on that lineage, 0.5% on that lineage, about 1% difference has arisen between those genomes. So that's pretty cool, because amongst those 1% differences are the key mutations that make humans human, that make us special. Okay. But 1% of 3.5 or 3 billion is still a rather large number of differences. And there are two ways in which differences can arise and go to fixation in these lineages, in any lineage. So here's a simple thought exercise. We've got an ancestor sequence, which is ATG, and we've got one of the descendant species has replaced the middle T with a G. So we have a difference between the two descendant species. ATG is different from AGG. Okay. So what does that mean? Well, let's think about this. So there must have been the mutation from T to G on the right-hand lineage. And initially, obviously, the mutation is at frequency 1. There's only one copy of it. But then over time, apparently, it's eventually gone until every member of the species, the descendant species, the AGG species, has that green G. So that obviously takes some time. So we're dealing with a process which starts with a mutation and then what we call the fixation process, where that mutation increases in frequency until it reaches 100% frequency, fixation. Okay, now what can fix a mutation? Well, there are two ways in which a mutation can go to fixation in a population. One of which is really boring, and it's called drift. This is a stochastic process, which is basically just generation to generation sampling error. So here we go. On the left-hand side, you've got the frequency. We're starting at low frequency, and these are just different iterations of what we call a neutral mutation, one which apparently doesn't matter. So its frequency in the population is not governed by natural selection, because natural selection doesn't care. It doesn't matter whether you have a G in that second position or a T. It doesn't matter. Natural selection doesn't care. So it's just sampling error that determines the frequency of the allele. And sometimes, look at this red one. Look at that red one. We just by chance, oh, there is a neutral allele that has gone to fixation in our population. So one way in which we can account for the green G is it was a neutral process based on genetic drift. So this is entirely stochastic. Or, and this is where it gets interesting, it could be a deterministic process involving positive selection. And that's, notice, and this is important, notice how much faster and more efficient, more directed, more deterministic, naturally, the selective process is than the slow, clumsy, directionless, vectorless drift-based process. So the challenge, and this is what I'm going to be addressing for the rest of this talk, is how can we distinguish between fixations, the differences between us and chimpanzees, which are there by virtue of genetic drift, versus fixation, the differences between us and chimpanzees, which are there by virtue of positive natural selection. In other words, what happened? The graph on the left or the graph on the right. Now, there are multiple ways in which we can attack this problem, and I can only give you a flavor of the kind of ways we go about it, but I'm going to start with a kind of a cool example. Convergent evolution. Convergent evolution is when natural selection in independent populations produces the same outcome. And you wouldn't predict if it's a random process, if it's a stochastic process, you do not predict the same outcome. You need a deterministic, a driver, if you like, to produce that same outcome. This is convergent evolution. And this is convergent evolution in dairy consumption. So this is a rather recent invention in our species, was the domestication of cattle, goats, camels, sheep, and so on. And it's actually a brilliant idea, because what it gives us is a lifetime access to dairy products. Now, think about what dairy products are. It's mammalian milk, people. Mammalian milk. Think of your mother's milk. It's the perfect food for a baby. This is perfect mammal food, is mammalian milk. And obviously, our ancestors, before we domesticated cattle, breastfed baby until baby was 2 or however old, and then baby, that was the last time baby tasted milk. Now we can drink ayran, we can drink milk, we can have dairy products every day of our lives. But it's required a big shift. The major component for a carbohydrate in mammalian milk is a sugar called lactose. And that has to be broken down in order to be useful. And there is an enzyme in mammals called lactase that breaks down lactose. Now, in populations which are not lactose tolerant, which is, by the way, all our ancestors were lactose intolerant, that gene is turned off at the age of 2 or so, because you don't need it. There's no point in producing lactase because you're not going to encounter any milk, right? But now with infinite dairy, we need to keep it switched on. And there have been mutations which keep the lactase gene switched on. And the cool thing is, this has happened in parallel, convergently, in 2 separate populations. So in an African population where pastoralization has arisen, in other words, we've got domesticated cattle, we have a particular mutation. And in European populations, and by the way, European domestication of cattle and so on occurred in, I believe, Mesopotamia. So it's the part of the world that you're living in, or close enough. We've got the same phenotype, the same outcome, which is keeping the enzyme turned on, but different mutation. So let's just explore that in a little bit of detail. On the blue on the right is the actual gene that produces the protein lactase, which, remember, digests lactose. And you've got a switching regulatory region in orange. And I've marked in cerise the switch, the bit which is actually involved in switching off in ancestral or in lactose intolerant populations at the age of 2. So let's zoom in. Okay, so we're just looking at the regulatory region. Now let's look here. Here in blue, we see a mutation which has disrupted that regulatory region in African populations, only in African populations, okay, to keep lactase switched on. Now, let's look at the European situation. It's in the same region. It does exactly the same thing, namely keeps lactase switched on, but it's different mutation. So that's not random, people. There's nothing stochastic about that. It's a cow, in case you've never seen one. That is the power of positive selection acting independently on these populations to result in convergent evolution. Okay, but that's kind of a special case. Can we have a more general idea of how we can distinguish between between drift-mediated fixation and selection, positive selection mediated fixation? And the answer is yes, through looking for what we call selective sweeps in genomes. Now, what do I mean by that? Well, think about a new beneficial mutation which arises. It's going to sweep through the population. It's going to increase in frequency, right? Now, the key thing is the timing piece. Remember in the animation I had earlier, the drift process to fixation is very slow and inefficient, whereas the selection process is rather fast and rapid because it's vectored, because it's directional. Now, what does that mean? It means that while that's underway, what we're doing is we're fixing a particular mutation. But what happens to the flanking genetic variation associated with that beneficial mutation on a chromosome. So let's start with a super simple example. So let's imagine there is no genetic recombination. So each of these chromosomes is effectively an independent clone. They don't exchange genetic material. And by the way, there are plenty of examples of regions even in your genome where that occurs. So there's no recombination. And now let's say, as you can see, the white A on the green chromosome is a beneficial mutation. So I've color-coded the chromosomes. We've just got a sampling of the variation. So we got a green, yellow, and a turquoise chromosome. So what's going to happen? Well, natural selection is going to favor the white A. Okay, so that's going to increase in frequency. What does that result in? What it results in is essentially the elimination of all chromosomes without the white A. So eventually the entire population will be comprised of individuals with the white A. That's what natural selection does, right? But notice, and this is the critical thing, the entire population is fixed 100%, not just for the white A, but for the flanking variation. In other words, the variance at those three other variable positions that happened by chance to be associated with the white A. So what does that mean? It means as we survey levels of genetic variation along a chromosome, maybe it's up, then suddenly crashing down, no variation, no variation, no variation, then it goes up again. What does that mean? It's this process. What is eliminated? That whole slab of genetic variation, a selective sweep of under positive natural selection. So as you see in the left, we've lost variation in the entire chromosome in the case of a non-recombining chromosome. But now, what happens if we are more realistic and we look at a situation with some recombination? So in other words, some genetic exchange between chromosomes. So again, same scenario. We've got the white A arising on a green chromosome. We've got three different chromosomes, the turquoise, the green, and the yellow. Now, this is different because during the sweep process, you can have crossing over events. So you're going to create sort of mix and match genetic variation. So it's not only going to be the green T in position 1, which is associated with the white A, but here we've got the turquoise C also associated with it. And obviously this can keep going. Let's have another recombination event down here. So you see what we're doing. We're creating different permutations of coupling between the white A and the flanking variation, courtesy recombination. But note too that the white A is next to a G, and because they're very close together, the probability of recombination, a recombination event, a genetic crossing over event occurring between those two sites is very low. So even though you've got mixing and matching further away from the target site, you're not getting it very close to the focal target site. So again, what happens? Well, we're getting selection is favoring the white A, but we notice we've got several different chromosomes. So again, these are going to multiply. Now let's look at the pattern we see. So here's the outcome of the selective sweep. Now we've got a more local area of reduced variation. But again, the footprint of positive natural selection, and obviously the size of the footprint is going to depend on the intensity of selection and the rate of recombination and many other parameters. But basically the message is this: there is a footprint immediately post selective event where you've lost variation in the population in the region around the key mutation, in this case, our white A mutation. And when we go into nature and look for examples, we find them. This is one I rather like. It's kind of a cool example. This is in the nasty bug called Plasmodium that causes malaria. We've been trying to kill malaria for a long time, and we have some anti-malarial drugs which try and take out Plasmodium. Bad news, Plasmodium has evolved resistance to the drugs we use, the anti-malarials. And this is a detailed genomic population genetic study of one of those regions, genetic regions in which resistance has appeared. What you can see on the frequency of variation, basically genetic variation in the population, is on the left is the vertical axis, and we're just walking along the chromosome along the horizontal axis, and you got normal level of variation, crash, very little variation, then it recovers, exactly as predicted by the model I've just given you. Namely, you've got intermediate levels of recombination, you've got strong natural selection in favor of a mutation in the heart of that reduced variation region. So, and we now know that there is indeed a gene which is involved, has mutations which are involved in resistance in Plasmodium to these drugs, right there in the center of that area of reduced variation in the genome. So that's one way, or a second way, convergence and sweeps. A third way is what I'm calling acceleration. And this is particularly germane to studies of our species in comparison with, say, chimpanzees. Sometimes you find bits of DNA which are clearly under strong negative selection. They're the same when you look across species, okay? Negative selection is keeping them the same. These are functionally important. This is a beautiful example. This is a piece of DNA. Look at the comparison between chickens and chimpanzees in the bottom of the table on the left. There are virtually no differences. So you've got eons of negative selection keeping that piece of DNA basically the same, just two differences between chimpanzees and chickens. Look at us, it's gone crazy. So you've got an important piece of the genome. We know it's important because it's highly constrained, but it's suddenly started evolving very fast in our lineage. That suggests that this is not a random event. This is not drift. This is positive selection. Another cool example, and you might have heard of this, this is called FOXP2. This is a transcription factor, so it's a gene that controls other genes, which has been implicated in medical studies for people who have problems with speech, both creating speech and understanding speech. And it turns out, yes, this transcription factor is involved in the construction of your brain. Turns out this is a highly conserved, lots of negative selection here too. Notice you've got, if you compare a rhesus monkey sequence of this gene with a gorilla, there are no amino acid differences. So highly conserved, lots of negative selection, but bang, look at the human lineage. Humans and Neanderthals have 2 amino acid differences. Now, it's only 2 differences, but still, that's really interesting. A gene that we have a priori reasons to believe is interesting from a linguistic point of view, and bang, it seems it's highly conserved across the animal kingdom until you get to hominins, humans and Neanderthals, and whoa, suddenly it's evolving. Again, positive selection. But the coolest area in studies of positive selection these days is with ancient DNA. Ancient DNA is so sexy. Ancient DNA is really, it's this new technology which allows us literally to travel into the past. So just very quickly, what is ancient DNA? Well, it's DNA from long dead things. And it's sometimes difficult to get. Sometimes the DNA is very degraded. Inevitably, DNA degrades after death. So the question is how degraded it is. So, you know, there are all sorts of technical problems. And only about 10 years ago did we have technological answers to those technical problems. So, I mean, very simply put, here's some DNA from a chromosome, and then you die, and what happens? Well, the DNA breaks apart. And so you don't have any nice long strands of DNA as you do with what you might call healthy modern DNA. What does that mean? Well, now, and this is the cool bit, we can go and sequence all those tiny bits. So every little fragment we've got a DNA sequence for. So that's what you might call brute force DNA sequencing. But next we need brute force computation, which allows us to cobble together all those tiny pieces, overlap them to reconstruct the genome of the long dead individual. And I'm sure you know this. This has already led to some remarkable insights. Here's a cave in southern Siberia called Denisova, where we had some good material, weirdly, from a tooth and a pinky bone, from a little finger bone. Managed to get genomic material, which has introduced us to a whole new relative. This is the traditional family tree of humans. We've got Beyoncé as our representative now. Here's our closest relative, Neanderthals. It turns out we have two closest relatives, but we don't know what the Denisovans look like. Denisovans are related to Neanderthals. They're quite distinct from the Neanderthals, but we only have a little finger bone and a tooth so far in terms of understanding what they look like. In other words, ancient DNA has contributed to revolutionizing our understanding of who we are and what we're related to. But this is the cool stuff. It's a time machine to help us study the fixation of mutations. So in other words, to directly address the question that has dominated this talk, when I find a difference between two species, is it there because it's a neutral allele that drifted to fixation in that lineage, or is it there because it's a positively selected allele that was driven by natural selection, because this was actually an adaptation? It was making the zebras run faster. It was making the spider's web more effective. So let's talk about what you might call the traditional approach to understanding the evolutionary past. So here's a family tree of two species, and I've got multiple populations in those species, and I've marked with the colored dots mutations. So our job is, we live in the present, and we can sample species 1, 2, 3, 4 and in species 1, and populations 1, 2, 3 in species 2, and then we can try and interpolate what happened in the past. So the fact that 1 and 2 in species 1 both have the green mutation, but 3 and 4 do not, tells us that the green mutation arose in the common ancestor of populations 1 and 2, but after the split with population 3. The fact that all 3 populations in species 2 have the purple mutation, but the purple mutation is not present in species 1, tells us that mutation arose in the common ancestor of all of species 2. We're working backwards, right? And that works, but this is where it gets exciting. We've got a time machine. Ancient DNA is a time machine. So we can actually travel back and look at populations way back then. Right? So now we can actually track through time. We can look at our favorite allele, the purple allele, let's say, and see what's happening at different stages and different populations as we travel through time. Ancient DNA allows us literally to track changes in allele frequencies. It's a time machine. And here's a super cool article, which was the first sort of concerted attempt to use this logic. So we're actually going to track the change of mutations through time by studying truly ancient, less ancient, rather recent, and modern populations. So, well, I mean, here's what we're doing. So what I've color-coded allele frequencies here. So we're starting way back when, say 20,000 years ago, and we got high frequency of a low frequency of the new allele. So we're going to make that a blue color. And eventually over time, we're going to get increase in frequency to the orange state. So that's just over time, the population is going from blue to orange in terms of frequency. And we're actually watching evolution unroll, if you like. We're watching it in real time, courtesy our time machine. So this is the data, and it's complicated, I admit, from this Matheson et al. Paper, but it's super cool. So what we're looking at is the human genome. So each of the chromosomes, all 22 chromosomes, as you can see, are laid out there. They're color-coded. What we've done is we've looked at genes the whole way across the genome. So each chromosome has a whole bunch of different genes that we've looked at. And what we're looking for are vectored change across those populations that we've sampled. So we've gone back to, I think, about 10,000 years ago, and we've sampled multiple populations since that time. And so the genes that are most likely evolving in an interesting adaptive way under positive selections are the ones which are going to be highly different, really altered over that period of 10,000 years. And you can see there's a dotted line at the bottom, which is a statistical threshold. So anything that comes above that threshold is probably, not necessarily, but probably under positive selection. And remember, these are the changes that matter, that make humans human, right? And this is a really nice proof of principle that what we have, the strongest signal we have, is from a gene we've already discussed, namely the mutation that confers lactase persistence on modern populations. So we already knew that, so we haven't discovered that, but it does suggest that this system is working really well. So that is a super cool way to unpick this problem of, was this brought about through a drift mediated process or through a positive selection mediated process? Let's get into a time machine and actually watch evolution in action. That's what this is. And the final perspective I want to give you on ancient DNA and adaptation is, so cool. Remember what happened. So it's not just ancient DNA, Neanderthal DNA has not just told us about the Denisovans being out there, different, if you like, unfamiliar, a whole new, maybe not species, but a whole new member of the human family tree. We've also learned something really amazing, that everyone out there listening to this talk who is not a pure African has in their genomes bits of genetic material which are derived from hybridization between Neanderthals and Denisovans and our ancestors. Homo sapiens, modern human beings, as they came out of Africa about 70 or 80,000 years ago, met these other populations out there in Europe and Asia. Neanderthals, Denisovans. Remember, the Denisovans, all we know about them, at least to start with, is based on this material from the cave in southern Siberia. And what happened? They had sex. What does that mean? That means the descendants of those hybridization events have both human, modern human, and Neanderthal and or Denisovan DNA. So as you now know, every non-African has, on the average of about 1% of their genome is derived from Neanderthals. Okay, that's cool, but doesn't relate to what I'm talking about. This is where it gets interesting. Turns out that we didn't just, if you like, hybridize with Neanderthals. We didn't just acquire sort of random bits of DNA from them. We acquired, when I say we, we're talking about non-Africans in general, Homo sapiens. We acquired all sorts of good stuff from these guys. Now think about it. Neanderthals are the product of a group that left Africa maybe 600,000 years ago, moved to Europe, Western Asia, okay? They then evolved under natural selection to cope with the new challenging environment in which they found themselves. They're no longer in the tropics, they're in miserable, cold, windy Europe. They're evolving rapidly to adapt, if you like, to that environment. Now, along comes our ancestors, hello Neanderthals, they have sex. So what our ancestors got was the adaptations. They got the genetic adaptations that the Neanderthals had, if you like, quietly evolved over the previous 500,000 years. Cool example of this. Tibetans, as you probably know, are remarkable for their ability to live at high altitude and breathe very low oxygen content air. So if you as a Istanbul sea level dwelling Turk, go up to the summit of even one of the big Turkish mountains, you go to Uludağ or Öşüzdağ, wherever, and you try and run up hill, you're gasping because there's just not enough oxygen to keep your muscles active. So it's a real challenge living at reduced oxygen at high altitude. But Tibetans are extremely good at it, and it's an inborn facility. And for a long time, people have investigated this, and they've identified a gene called EPAS1, which is involved in oxygen metabolism, which the Tibetans have a very different version of from the rest of us. So that's the trick. That's the Tibetan trick to adaptation to high altitude, is this particular very distinct gene. We now know, I think this is so cool, that where they got that very distinct gene from, a gene with a whole bunch of distinct mutations which facilitate living at high altitude, breathing the thin air, surviving with low oxygen. They got it from Denisovans. So as you can see in the family tree at the top, you've got a common ancestor, you've got this gene was evolving, but then you've got a horizontal transfer. You've got that arrow from Denisovans to Tibetans. Amazing, okay? That's, talk about a positive acquisition from your neighbors. But there was a problem with this idea. So this was an observation we made from comparing genomes between Denisovan genomes, Tibetan genomes, non-Tibetan genomes. But there was a problem. The cave, the Denisovan cave, is at fairly low altitude. So why would Denisovans be adapted? Why would they be adapted to living in low oxygen environments? Just a year or two ago, we found another piece of Denisovan material. It's a lower jawbone. So now we finally have a bit more Denisovan material. The really cool thing is, where did that jawbone come from? It came from a Tibetan cave at over 10,000, at over 3,000 or 4,000 meters altitude. In other words, the Denisovans were living at high altitude. So yes, they were adapted to life at high altitude. And yes, that is the key to Tibetan adaptation. There is a beautiful instance of positive natural selection, a wonderful and critical adaptation for life at altitude in low oxygen environments. Okay, folks, that is all I have to say. Just a reminder, if you've put questions in the chat, that's great, and do continue to put questions in the chat. If, however, you would prefer to connect more privately, so to speak, I've just put my email address, berry@oeb.harvard.edu, down there at the bottom of the screen. Okay, I will put my spectacles on and see what we can find in the chat. I'm going to kill the screen share. Okay, chat. We'll be starting in 3 minutes. Is that the only question that I have provoked? Will be, and it's not even a question. No questions, maybe because I explained everything so brilliantly clearly that nobody, no, here they are. Thank you for saying Alper Karadenis. What is the most fascinating thing about evolution for you? Oh, that's easy, us. We're extraordinary. I mean, we're just, and remember what we are, and this is what we've learned from Darwin. We, brilliant, smart, brainy, us, are modified great apes. Modified great apes. This great brain is a modified ape brain. So that, for me, I think that realization is one of the most sobering and seismic ideas ever in the history of ideas. One. Two, I think what's exciting now is we can use the kind of tools that I've just described, looking for selective sweeps, looking for convergent evolution, looking for using ancient DNA to travel in time and try and spot the key genetic inventions, if you like, that make humans human, i.e. distinct from Neanderthals, distinct from chimpanzees, and so on. So yeah, I don't want to be, it's a bit boring, isn't it? Say the most important thing about evolution is us, but I think we're allowed to say that because we're humans. So of course we're interested in humans. Salikaya says natural selection immolates the ones with weaker traits, but it also reduces the diversity of the gene pool. So can this reduction in the range of the gene pool harm the population in the long run, or is it beneficial to the population? Interesting question. Remember what natural selection is doing in it's eliminating deleterious mutations. So by definition, they're the problem mutations, so you want to get rid of them. Okay, now there are situations, and so I showed you the situation of a non-recombining chromosome under a selective sweep. There's positive selection has effectively eliminated genetic variation. Now, presumably that sweep wouldn't go to completion if it was coupled with increasing the frequency of a deleterious allele. So I don't think that even strong positive selection is going to probably result in the fixation of highly deleterious alleles. Yes, you are going to lose some genetic variation. Genetic variation comes back, people, because mutation happens. Mutation is inevitable. And there's no sort of intrinsically important reason why a population has to have a lot of genetic variation. It's useful in terms of responding to future adaptive challenges and so on. But it's not like you have to be a viable population, you have to have a certain level of genetic variation. The problem with a loss of genetic variation is when you're in a particular situation where you've got a very small population and you get a lot of inbreeding. So this is inbreeding depression. The problem then is that low frequency deleterious mutations are likely to become what we call homozygous. In other words, you get two copies of them. They're recessive, so normally you don't see two copies of them, so they're not expressed, but in those situations they are. But that's a particular demographic situation where you have a small population and little opportunity for breeding outside your immediate group. But it's a really interesting general question, is what are the factors governing levels of variation that we see in populations as a whole? Bengisu says, which discovery in biology has made you most excited? So now we're moving, we're moving beyond the big discovery of evolution. What do I think is the coolest thing in biology? You can't get away from the double helix, I'm afraid. I mean, it's again a rather boring and simple answer, but it's just such a stunning idea. Remember, before 1953, it was still that people weren't even sure that DNA was a hereditary substance. You have this one simple idea that you've got this anti-parallel pair of strands, and you've got this simple 4-base code, and bang, 1953, so many doors have opened, so many opportunities to do science, to understand the process of life. But from not understanding how we're encoded to having a really remarkably, it's not complete, but it's much more complete. That was a key inflection point, I would say. Interesting stuff. This is Arif Kemal. Is it necessary Denisovans lived at high altitude to have a gene to cope with this condition? That's a really interesting question, and I think the answer is yes. Let me explain. You might think that natural selection, if this is a good variant to have in some circumstances, would support that. But I'll give you a thought experiment. Let's take two islands and let's have a population of zebra on island A and on island B, and they're the same zebra. And on island A, we have lions. On island B, we have no lions. Now, what we'll see on island A is obviously there's an advantage to zebras that run faster. And they will be less likely to be eaten by lions, so they will leave more offspring and so on and so forth. So you will get evolution of increased running speed on island A with the lions. Island B, there's no advantage to running faster. Okay, so there's no but you might say, well, there could be an advantage because lions might arrive. No, natural selection isn't looking to the future. Natural selection is just we've got a bunch of individuals here, some which run faster than others and some which run slower. It doesn't matter. So on island B, running speed is effectively a neutral trait, so natural selection would ignore it. And the same logic applies to being able to breathe oxygen very efficiently at low oxygen tensions for the Denisovans. If you're living at sea level, there's no advantage to having that mutation. So you would not expect it to go to high frequency in the population. Let's see, where are we now? Hamid Kartal asks, do you have any unforgettable memories in the lab which you could share with us? Okay, I do actually. So the reason I like talking about selective sweeps is this idea that you lose variation in a particular region of the genome if natural selection has, positive natural selection has affected it recently, is a long time ago, it is a long time ago now, I was arguably and people might debate this, I introduced the term selective sweep and discovered the first good example of a selective sweep, which was working in fruit flies because they were easy to work in. There turns out to be a small chromosome in fruit flies which does not recombine. So that whole chromosome is effectively behaving as a single unit. It turns out it's more complicated than that, we now know, but at that stage, that's what we thought. It was a single unit, it did not recombine. So if you've got a positively selected mutation, so imagine you've got 400 different copies of that chromosome, each with different neutral variants, but then on copy number 98, you get a beneficial mutation, which is driven to fixation. That means the entire population at the end of that process will be fixed for chromosome 98. Everyone, we've lost all genetic variation on that chromosome. So I do remember, so we did some work on this chromosome in fruit flies, thinking it would be a good place to look for a lack of genetic variability. I still remember, so in those days, you didn't have automated DNA sequencing. Yeah, God, it was hard. In those days, you had to do manual sequencing and so on. You put the sequencing process on a big piece of photographic, it's X-ray paper, X-ray film. You put it, you develop it. So you don't know what's happened until it slowly comes chug, 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 chug out of the machine. And then you can glance across, and I've got 20 different individuals of fruit flies from around the world, all for the same piece of genetic sequence from the same gene on this non-recombining chromosome, and bang, they were all identical. It was amazing. It was actually super cool. We weren't sure it was going to work, but yes, that was my. It's sort of boring, isn't it? But that was my sexy lab experience. Selma, we found out how Neanderthals looked like by finding their skeletons. Yes. For example, in a crime case, can we find the exact look of the culprit by using, oh, that's a really interesting question. Can we basically figure out what an individual looks like from a DNA sample? Highly controversial, but yes, to some extent, but not to a major extent. So for example, there are plenty of examples of identical twins who obviously by definition have very are genetically identical. So, and yeah, many identical twins do look very similar, but occasionally you'll have two identical twins, one of whom, let's say, had a disease as a child, and you wouldn't even know they were brother and sister or brother and brother, okay, because they look very different because of that experience that they had. So then you've got this problem. So I've got this DNA, but I can't really make a strong extrapolation to what they look like. There's certain characteristics that you can tell, things like eye color, hair color, aspects of complexion and so on, but nothing really. So, okay, so you can probably say that whether it was a white person or not a white person, but you probably couldn't make statements about, they had a strong jawline and a big nose or whatever. So rather limited, given the effect of the differences in environment in terms of our upbringing. Think of the identical twins, which ended up looking very different, but really interesting question. What is the possibility for us to discover other extinct hominids, Denisovans, in the future? Oh, I think very high. I think, again, really interesting question. there are two ways in which we can do that, which is one is archaeologically. So we're getting, or paleoanthropologically, we're getting better and better at finding bones material. We're getting better at getting a better systems for looking for material and so on. So, and look, we've had several new species of hominin. So hominin is a group from that 7 million year ago point. That's the common ancestor of humans and chimpanzees. Then you've got a lineage which is leading to us. All the species on that lineage and all branching off it are the hominins. And we've discovered several new ones in the past 10 years, including, well, 15 years if we include, there's this famous tiny species called Homo floresiensis, which is found in Indonesia. And just 2 years ago, we found another dwarf small individual species in the Philippine Islands called Homo luzonensis. So, and that's all from just traditional osteological from bones. But I would imagine there's a good chance as we get better at ancient DNA and as we get, as we have more opportunity to explore ancient DNA worlds, so to speak, that we will find new relatives. Now, ancient DNA will only allow us to go back about 1 million years. So there's not going to be that much in terms of from DNA analysis, because probably most the new stuff we'll find will still be archaeological, which is earlier than that. DNA degrades inevitably, and it's not quite a hard time limit, but pretty much once you reach 1 million years, you're probably out of luck in terms of actually being able to get any usable sequence. But really, really interesting question. And Yassine, doesn't evolution tend to work in a way that reduces mutation? If so, does that mean that evolution gradually slows down? So in a way, evolution reduces mutation in that negative selection eliminates mutations. Neutral, drift-based will either go to 100% or 0 eventually. So again, you're losing it. And a selective sweep obviously is eliminating variation because you're replacing existing variation with just the one type that you've swept through the population. But remember, there's an input the whole time as well. There's this mutation process. Remember, I started with that slide that every single one of you and me have about 60 or 70 new mutations. So yes, there are evolutionary processes which are removing variation, mutations from the population, but there are also processes, namely mutation, which are feeding mutation into the population. So no, I don't think there's some sort of will inevitably sort of slow down and become less and less variable, because mutation is essentially a thermodynamic inevitability. So despite all the evolution to try and reduce rates of mutation, try and eliminate copying errors in DNA replication, it happens. Think of those 60 or 70 differences between you and your parents. No, it's going to be a constant sort of give and take, elimination of variation through selection, through drift, but also then the injection of new mutation courtesy the mutational process. And let's see, that I think is that. Oh, hang on. Denise has come through. How was the RNA world theory theorized if DNA degrades in about 1 million years? Oh, interesting. Yeah, so we don't have any actual sort of time machine data on the RNA world. Very briefly, the reason we believe that life originated as an RNA is there's essentially two things. One is, given the fact we store information in DNA, and given the fact that the active component of living systems is proteins, why would you involve a secondary molecule called RNA? And remember, RNA is embedded in all sorts of ways: messenger RNA, ribosomal RNA, transfer RNA, and so on and so forth. So that's the first reason. The fact that you've got RNA performing all these critical basic functions in living systems suggests that it must have been integral to the system from the start. The second reason is a more conceptual one, which is there's always been a sort of what you might call a chicken and egg problem with the origin of living systems. Namely, you need to be able to encode information, DNA sequence, let's say, but you also need to be able to enact, to make that information active. So those are enzymes, those are proteins. So you need proteins to make DNA, but you need DNA to encode proteins. So how do you break that cycle? Which came first? You need them both. Okay, then we discovered, and this isn't that long ago, about 25 years ago, we discovered that RNA, unlike DNA, can act as effectively as a protein. It can act as a catalyst. It can act as a so-called ribozyme. So remarkably, when I say we've got a chicken and egg problem, DNA protein, RNA solves that problem because it's both chicken and egg. So its sequence can contain information, but it can also auto-catalyze, so to speak. So it can act as a so-called ribozyme as well. But it's obviously highly speculative stuff, but really interesting. Okay, any final words? In that case, thank you very much for listening. I hope it was at least somewhat illuminating. And I want to say a big thank you to Dennis and Jeren and anyone else who was involved in organizing it. Really appreciate all the work you've done. And thank you guys for listening. And I'm hoping to get to Sabanji at the end of the spring semester. So I hope to see you in 3 dimensions. In a few months' time. All right. Thanks, guys.